Okay, what about now? Is it? Okay. Um, that's annoying, but whatever. Okay. It's working now. Hey guys. Hmm? Oh, it's the Peter Kittner video. Okay. So, so you can get this on. Yeah, okay. Not with the shirt I'm wearing. Hold on. Here, I'll put something. Let's see if it just works right out of the bat. It'd be cool if it actually did these because I have a folder just full of MIDI files. So I'm gonna try that real quick. Oh, I have it as the Super Mario Bros. 3 thingy right now. So here's what we're gonna do. We're gonna change the sound font. So give me a second. It doesn't sound bad. I just think it's really funny. Let's see. Let's do the default, or here, we have a Yamaha, or let's do the FreePads. This is what, if you're wondering, this is what flat.io uses. Apparently not. Because this does not sound, it sounds worse than I thought it was going to, actually. Let's see. I think it's this one. Maybe? It is, yeah. This is what flat.io uses. But, actually, I don't want to listen to this. We're just going to listen to regular music. All right, we're going to pull up. I'm going to pull up the best. Oh, yeah, let me fix the chat thing. Chat dialogue real quick. Hold on. It was the best of times. It was the blurst of times. Let's actually put this on for a moment. Not that one, this one. This is peak.
It was the blurst of times.
You actually couldn't hear any of that because my thing was muted.
It was the best of times. It was the blurst of times. It was the best. You stupid monkey. It was the best of times. It was the blurst of times. It was the best.
There's a weird appeal to that, you know? I don't know how to describe it. All messages you send will appear publicly. You know how we're gonna fix this? We're just gonna hide that section. Boom. Oh. It's almost like it doesn't exist. But anyways, let's get this music rolling, and then we can get locked in. I'm just gonna throw all of this in here. And this too. And maybe some, we could do, let's see. Sorry, I'm just deciding. Music is very important. We'll do Super Mario RPG. Let's actually start with that. Never mind. Alright. Let me get my biology textbook. Actually, let me change my shirt. Let me get my biology textbook. dokie. Ooh, this is fire. I'm gonna check the audio levels real quick. Blah 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 blah. I think I'm loud enough. All right. There we go. Okay. So you'll just see me reading a textbook. I'm going to be actually my notes, not a textbook. Yes. And actually, see if I can make this happen. This isn't. Okay, I need to shut up. Hold on. Thank you. Okay. So what? Oops. Okay. Why is it flat? You hear it, right? Like, it's gone flat. You can't just pretend that didn't. Okay, well, but anyways, we're just gonna be reading this, and then I will be pulling up the slides relevant for each chapter on here. So that's what my bio teacher likes to put these like memes on here, and so that's what I'll be doing. You can also yell at me if you can see that I'm getting on my phone, and that was the exception because I'm just finishing one last thing. Oh, we're two subscribers away from 600. That's cool. Okie dokie. Let's lock in. So we're going to start with chapter 1. Notice how many pages there are. That's how many slides we've watched over the years, or over this entire year. I see. There we go. One does not simply walk into AP Biology and get an A. This is true. You do sometimes have to do the work on your own. Okay. I'll just move the slides along, but you know, some of these first terms are very basic. You know, a 3-year-old would know what these are, but not me apparently, because I absolutely bombed my first test. But that's okay. All right. So biology is the study of life, and then so cell is the most basic unit, of course, and there's different levels of organizations. Right. So you have. Are they not here? Whatever. He probably just said it in person. But basically, it's the atoms, to the molecules, to cells, to tissues, which cells make up tissues make up organs, and then organs, you know, they're part of their organ system, which is all part of an organism, and then multiple organisms make one population, multiple populations make a community, multiple communities make an ecosystem, and then, you know, all the ecosystems make the whole biosphere, which is basically just, you know, the land masses on Earth. Why is it called Chrome? Alright, we're going to rock out to this. But then there's 2 types of cells. Not what I meant to do. There are 2 types of cells. There's eukaryotic, which is basically animal cells. If you want to think of it that way, it's more complex than that. But there's 2 kinds, right? There's the, what do you call it? Well, there's 2 kinds. There's eukaryotic and prokaryotic, and then this is the animal cell, so it has a nucleus, and it has membrane-bound organelles. prokaryotes don't. But what they both have is they both have a membrane, and then they both use DNA as genetic information. It's just the way that they hold is a little different for both the kinds. Then we have unicellular and multicellular organisms. Can you take a guess what that means? I'm not moving on. Or I'm not. Yeah, you got that. All right, and then life is classified into 3 domains. This is what I absolutely did not know, so I should probably double check now. That there's bacteria, then they're prokaryotes, right? Then you have archaea. I don't know how to pronounce that. They're basically old types of bacteria. And then you have eukarya, which are plantae, animala, fungi, protists. It's multicellular organizations, basically, or organisms. That's basically what it is. Just so you know, that's one page, and I have written all the way up to here. And apparently, what my friend has done is she's filled out an entire book. Hello? What do we have to write? But that's besides the point. We're just gonna keep moving on. Alright. Okay, so here, the 6 characteristics of life: grow and develop, respond to stimuli. So you have to be responsive. You need to reproduce. We'll talk about that in a much later chapter. You have to obtain and use energy somehow. So, like for example, this doesn't use energy; it's abiotic. You have to have a genetic code, because how else are you going to, how are you going to make yourself? Because DNA codes for proteins, and proteins do almost everything. Then you need to have cells. Duh, it's the basic building block. Atoms make cells, cells make tissue make organs, and an organ system. The whole thing. What the freak? Holy fold. Okay. Homeostasis is the ability of an organism to regulate a stable internal environment. So think of it like this. The examples are right here. It's easier when you don't. I'm not going to explain all this, otherwise we're going to be stuck here. It's been 15 minutes already. But basically, when you're hot, you sweat, and then the water helps cool yourself down. We'll talk about water's properties in a little bit. And then you shiver when you're cold. Because, movement helps produce heat. Evolution is the change over time, central thing. It's the dogma of biology. Nothing in biology makes sense except in the light of evolution. So these giraffes, they actually, there's a funny story behind that. We'll talk about that once we get to evolution. And then you've probably seen this mice in like 8th grade. I would hope you have, at least. Otherwise it'd be, weird and stuff. I'm going to skip over some of the easy terms, and we're just going to move on to the harder ones. Hypothesis. Please know what a hypothesis is. If you're this far into your life and you don't know what a hypothesis is, you've probably never been to grade school. Okay, that helps. I will turn the mixture warmer. Actually, no. It will make it warmer. Not that you can really see my camera anyway. See, it's just blurry, regardless. But that's fine. The whole point is just to show that, like, I'm reading my biology textbook. But anyways, let's continue. Okay. If you're in stats, this is gonna get drilled into you a lot. Null hypothesis is basically saying there's no significance between expected and observed data. And AP Bio, we specifically use the chi-square tests, basically, chi-square stuff. And then our alternate hypothesis, it could be H1 or it could be HA, is just the hypothesis the researcher is trying to test, which is a little backwards from psychology, but whatever. Controlled experiment, variables unchanged. You'll learn about this in Psych too, AP Psych. Basically, there's a whole bunch of ways that you can tell if it's an experiment or a correlation or a faulty experiment or meta-analysis. Alright, so a fact is an observation that can be repeatedly confirmed. Bell rang last period. That makes sense because our school has bells. Maybe not to you, but Positive. Okay, so this is what I get confused with all the time. So positive control group, basically. So the experiment defines experiment with definite results from the variable. we're gonna look this up. Versus negative control group. Positive groups are to ensure experiment works by using a treatment known to produce a positive result. Negative control groups use a placebo known treatment to identify false positives and confirm the experiment results are caused by the variable. Okay, so negative helps control for the third variables or confounding variables, and then positive just confirms that, you know, the variable in question is actually working. I forgot what he was asking with this one. Independent variable, you know, it's what you change, and the dependent variable is what you are, you know, looking for, because it's dependent on the independent variable. The independent variable, in the, by its name, its results are independent from other factors, hopefully, and then the dependent variable, however, is dependent on other factors. In this case, the independent variable, if it's a good experiment. All right, let's see here. Theories do not become laws with more evidence. So theory is the how and why, and then the law is the what. It does not explain how. So the theory of gravity and the law of gravity. Interesting. There's an onion. You can't prove a negative. All right. I hope you know the SI system, and everyone knows base 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's not like we're doing, you know, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32. That's base 2. Alright. Length. Mass, temperature. Please, I hope you know what those are by now. Alright, now we're going to get to the hard stuff soon. This is true. Energy flow. Alright, we'll talk more about this later. You know, light energy is changing the chemical energy. Look at this. This is a plant. We'll talk about this later. And then, you know, recycled. Like, there's the nitrogen cycle, all that sort of thing. And then every trophic level loses heat. It's only expected that you keep about 10% of the level below you. Controlled experiment. Basically, you're just supposed to know what these are. I forgot if we ever actually talked about error bars, but basically, I'll show you an example. That's what you think they are. You're given a standard deviation, and basically you just say, like, you know, it's this far away both directions. That's what it is. It makes more sense if you have like a piece of graph, but see, notice how it's the same. So you have your average value, and then plus or minus your error. Because then, you know, you're giving yourself a range. Because if you're 100% confident, then you realistically you would need all the data, but usually you're not given it. So then you have room for error. Let's see. Oh, sorry. Oh. I just wanted to... it goes off screen. I'll make it prettier later. But, okay, for now, I'm just gonna move on. P-values, I could talk about that if it comes up. So, process of evolution drives diversity. Yeah, biological systems utilize free energy, molecular building, dynamic homeostasis. Living systems store, retrieve. This is DNA, genetic information, biology system interact. Organisms that are close to each other interact with each other. Alright, chi-squared test. You will learn this in stats, but basically, let's see if we yeah. So this is your observed. So let's say you have an observed value, but then you also have an expected value. So what you would expect to happen. In some cases, it would be like, you know, I expect, like for example, we did a worm catching experiment. And then so maybe your null is that I expect each color of worm to be picked up the same amount. But then maybe your observed says, like, you know, the ones that are more vivid color get picked up more often. And then so you do observed minus expected squared, and then you do that over e. And then this is sigma. This just means this, it's the summation. So basically for every different term, so like for each color worm, you're going to do this formula, and then you're going to add it all together to get the test statistic. Right, here's the p-value. Measure of strength of evidence against the null hypothesis. So, and it's going to be different in biology. Or actually, no, it's the same both ways. So a smaller p-value means that the test is significant. So usually the significance level will be 5%, or alpha equals 0.05. And then if p-value is less than that, you can basically say, like, there's a very low chance that getting this value or any more extreme values was due to chance. So basically it's saying, like, it's unlikely that this was an accident. Larger p-value is the opposite, as you would expect. See, if the p-value is less than, but then this is where it gets backwards, because, okay, so the way stats works, usually you say, like, for example, let's bring up a proportion test, one sample proportion test, right? And then so let's say our null hypothesis would be, like, for example, that the proportion is our claim, so it's what we're claiming it to be, and then our alternate hypothesis will be it is not what we claim to be. But then in biology, it gets switched. So the p-value is basically saying it's not what we claim it to be, but the alternate hypothesis is that it is what we claim it to be. And then so that's why these two would switch. So if the p-value is less, you reject the null hypothesis, but then if it's greater, then you fail to reject the null hypothesis. That's basically what it is. And then this is what I just said. You're given this table on your formula sheet. Don't memorize it, but remember your degrees of freedom, how to calculate it. Get the total amount of, like, different categories you're checking, because this will probably be categorical data, and then subtract 1. So let's say if we're talking about the worm experiment again, you have 6 colors of worms. Your degrees of freedom will be 5, because you subtract 1 from the total amount of categories. See, right here, number of outcomes. Critical value is the cutoff value. So, like, for example, if it was 5, we look at this value, and then if it's greater, then we reject, but if it's less, we fail to reject. I think that's what it's saying. Okay, this is probability, blah, blah, blah. You don't need to know this, it's fine. There's no significance between the expected and observed. So, have you heard the latest stats joke? Probably. Do you get it? All right. Now we're on to chemistry. Actually, we skipped in my class. We went straight to ecology. And I think the way is because, he did the units in a way where they sort of, we get most of the foundational stuff, because then as we get to further units, we learn new stuff, but then we also see this old stuff. So learning it afterwards wouldn't really make sense in his opinion. And then I agree with the way he taught us. I'm gonna drink some Gatorade real quick. All right. Ecology is the scientific study of interactions between different organisms and between organisms and their environments or surroundings. Ethology. Really? Okay, so that's the study of animal behavior, I guess. And then behavior is what an animal does and how it does it. It has both genetic and environmental factors. You can read the slides. I'm not going to read off the slides. All right, then I hope you know what the A prefix means. It usually means not. But here we have biotic factors, so basically they're living factors in an ecosystem, but they include dead things because they were once alive. And then you have abiotic factors, which were always non-living things. So like a rock. Rocks were never alive. They're pieces of stone. You probably heard these before. You have producers, and you have the everything above the producer is a consumer. And they either go through photosynthetic or chemosynthetics, that sort of thing. Consumers, heterotrophs. So you have autotrophs because they need to make their own energy. You have heterotrophs because they need to eat others for energy. You should know these. Oh, it was a seal. I'm pretty sure I've never met a herbivore. All right. This is important to know, so you can just look at it here. So this is an individual. It's an organism. But then multiple organisms is a population, and the multiple populations living amongst each other is a community. And an ecosystem is made up of both. It's made up of both biotic and abiotic factors. Like water is not alive. It's essential for life, but itself is not alive. Then you have a biome, which are multiple ecosystems. Probably. Then you have the biosphere. You know, it's called. Oh, actually, I lied. Biome is multiple communities, and then you know, flora, flowers, fauna, animals. I'm pretty sure. Okay, now this is some new stuff that I did not know before I took this class. Primary succession. Plants and animals invade where soil has not been formed. Secondary succession. It occurs when existing communities is cleared by disturbance, but that leaves the forest intact, like an abandoned farm or forest fire. And then, all right, we have energy flow next. I'm not listening to Friday Night Funkin' right now. Alright, I actually made this one. I didn't make the song itself, but I made this cover. Alright, so the food chain, you've probably seen it. This is a food web, but food chains, like for example, if we just went straight from leaves to Everglades, to the pig frog, to the alligator, but then so you can see the web is totally interconnected. And then so the arrows point where the energy goes, basically, and then at the top are the apex predators. Yeah, not much else here. Primary consumers. Okay, so basically you start at the primary producer, and then everything that eats that is a primary consumer. And then everything that eats them becomes secondary, and then you have tertiary, and then and then they can be multiple depending on who and what they eat. Decomposers and detritivores. I don't know how to pronounce that. Okay, so here's what I was talking about, the 10% rule. So you see, like, for example, these have 10,000 joules, so there's 1,000,000 joules of sunlight. But then realistically, you're not going to get all sunlight. Sunlight is inherent, it's also heat. And then so plants, they'll have 10,000 joules, and then when the herbivores eat them, but they'll lose about 90% of it to heat. And then so they'll only have 1,000 joules to use. And then carnivores, they'll only have 100, omnivores, they'll only have 10. So really, this is proof that you should be eating salads all the time. Alright, biomass. Total dry mass of all organisms in a given area decreases as you move up the chain in trophic levels. And I guess that makes sense. Okay, think of endotherms. The way I like to think of it is Five Nights at Freddy's. I have a point. Endoskeletons, right? They are a skeleton, so they're on the inside. So you think of endo as in, you know, they maintain their internal temperature, but it's inside. That's what endo is. And then exo, so if you think of an exoskeleton, it's like, you know, sometimes those stunt people, they'll wear, or some, like, people who have, like, injuries on their limbs, they'll wear, like, you know, like a brace, or maybe they'll have something that helps them walk. That's an exoskeleton. So it's on the outside. In this case, it's ectotherms instead of an X. It's a CTO. Ectotherms, and so they regulate their internal temperature from outside sources rather than internally. Biodiversity. It's life diversity. Just listen to Mario. But that's basically what it is. Oh my god. I wish I could get the text to rap, but I don't know how to do that. So, sucks to be me, I guess. If you guys know War Owl, I used to watch him a lot. I might watch him more. Biodiversity, you can expect, I guess. You know, the more polar you go, the less biodiversity you have. Makes sense. Extreme weather. All right, ecological niche, not niche. Niche. It's the role a species plays in its surrounding area. So, like, for example, you can see includes type of foods it eats, where it lives, here, where it reproduces, and the relationship with other species. Okay, so there's a vine. This niche empty feet, and then so he gets feet, but on here, it's this.
Hey Terry, you want some? This bitch empty. Hey Terry.
That's the vine. It's a meme. All right, survivorship curves. So it shows the number of proportions of individuals surviving of each age for a given species or group. You can also think of it like this too, depending on the. So if you think about it, is the goal to have as many children as possible, or is the goal to have quality children? And it can also depend on lifestyle, because I think we'll explain it here. So type 1 curve is organisms with low death rates in young and middle age, but then high in old. So like humans, because we typically, depending on the country you're in, population pyramids, AP Human Geo, you tend to live at least nowadays. I think average age is like in the 70s and 80s. And then type 2 is constant death rate. Apparently these guys, it's linear, I guess. Probably not perfectly linear, but And then type 3, high death rate in young, and then constant rate. So you can think of it like this. Plants have millions of seeds. Dandelions, those things that all fly off and when you go, those are all seeds. And then so you can expect that probably not all of them get planted, and that's what this is. They have a lot of seeds, and the chance that a couple survive, and then they reproduce to be these, they grow up to be these strong organisms that become resilient to its environment. Alright, you can probably guess what this is. Clumped populations are, what do you know? Clumped! And then uniform, it's antagonistic interactions. Sure, I guess. And then random is just unpredictable, it's not common. Pikachu, sorry. So density dependent factors. Would you like to take a guess what that means? Yeah. So they're, let's see. These are factors that depend on the density of the population. So for example, if there's more population, resources go down. If the resource amount stays the same. And then you have density independent factors. So like there's stuff that even if the population increases, it doesn't matter. So for example, natural activity usually doesn't care if there's one organism or if there's multiple organisms. A storm won't just not strike you because there's 100 people living there, I remember I was not paying attention during this one, but let's see. Proximate cause, how a behavior occurs, where it's modified, so proximate, how a behavior occurs, why a behavior occurs. Interesting. Okay. I didn't even write that much. I'm speaking a lot, but there's not much to write down. Okay, so innate behaviors. Innate, you know what it means? It's like it's like instinctual. They're fixed based on what your biology. Strict eyesight stimulus. So in this case, you can, like, for example, there's this thing called let's see, hold on. Oh, instinctual drift in AP Psych. So basically, we learned about it in conditioning. You can condition an animal, maybe to like for example, an example question is like, you can train your cat not to chase your mouse, your pet hamster. Let's say hamster. But then sometimes, even though it's learned not to, it may still. And then so, that's called instinctual drift because it's the cat's instinct to... I have a history with this song, so we're going to skip it, but that's okay. We're not going to do all that. Basically, but if I go back to my example, the cat may sometimes go back to chasing the hamster because it's its natural instinct. Cats hunt mice, gerbils, rodents, that sort of thing. So taxis, what is this? Change in direction, automatic movement towards or away, in positive taxis or away from the stimulus. Okay, so I guess what I'm getting from this is that taxis is very intentional, and kinesis is just kind of, doing it until you find it. Okay. Migration, you've probably heard of it when you've seen birds. We have crows. I think that just they like to fly in groups every now and then. Or maybe it's not crows. We have birds that fly. Organisms communicate to exchange information. Animals use variety, auditory, some communication includes dominance, delineate territory. Okay, so basically, animals communicate like we do, not necessarily the same way, but they do communicate. Signal stimulus that causes a change in behavior based on animal communication. So pheromones, I'm thinking of a Friends episode right now. When you you smell someone else's hair, I think that's a kind of pheromone, if I remember correctly. But then I guess you could also say if you smell and the hair smells nice, you can assume something, and if their hair smells bad, you can assume the other thing. That would be my guess. Let's see. Yeah, humans have pheromones, so I'm pretty sure it is through the hair. Okay, visual signals. So certain animal, like for example, poison tree frogs. Tactile. Male fruit flies. I hate fruit flies, by the way. Auditory signals. Okay, so I assume like bird calls. I learned behaviors. What do you think they're behaviors that are learned? What? Based on specific experience. So this one is, yeah. So cognition, you know, has to do with the brain. So it's the way you think. It's the way you interpret your surroundings and how you respond as a result. Associative learning. This is operant conditioning, I believe. Imprinting. This is just a natural thing, you know. So when I believe when some animals are born, they will immediately, you know, begin to follow and trust the first organism that they see. Usually it's their mom. I think sometimes you can be seen with humans. Spatial learning. I wonder what this example is. If that's a bee, I hope it dies. Or wasp, sorry. If it's a wasp, I hope it dies. Social learning, you know, you learn, you know, monkeys are very sociable animals. Habituation. We learned this in AP Psych too. So habituation is basically, you could think of it like this. Let's say you live by a train station. And then so the first few times you hear a train go by, you're shocked, right? But then after a while, you just become dull to the sounds of the trains coming by. Okay, let's see. Imprinting. So learning and innate components. This is, yeah, this is what I was just talking about. Cognitive map. We also learned this in AP Psych. So basically, you, by kind of, what do you call it? By interacting with your environment around, with the environment around you, you start to build this, you know, internal map where, you know, you can sort of picture things and you know where things are relative to each other. Associative learning. This one, like for example, I believe this specific example, I wouldn't say, I guess there is classical, but I was thinking more of operant conditioning. Pavlov's dogs. You know, you probably heard Pavlov's experiments. Basically, dogs salivate when they see food. And then what Pavlov did was, every time the food would be shown, he would also click the bell. And then, you know, the dogs are salivating to the food. The unconditioned response, the unconditioned stimulus is the food, and the unconditioned response is to salivate. And the neutral stimulus is the click. But over time, as he kept doing that, you have to play, I think you have to do the neutral stimulus before, or at the same, I'm pretty sure it's before you present the unconditioned stimulus, because then the neutral stimulus will become the conditioned stimulus because every time you click it, then the dogs will start to salivate. Apparently my psych teacher did that to the kids in our class. I wasn't there for that, unfortunately, but sounds fun. And the opera conditioning, it's basically, it's conditioning through behaviors and actions and that sort of thing. Mitochondria is the power. Funny you mentioned that. We're not supposed to say that. Mitochondria is where cellular respiration occurs. We're locking in though, don't worry. Cognition, process of, it's your brain, you know. Maybe psych. Learning by observing others. Social learning, it's self-explanatory. Agonistic behavior. I thought it would be antagonistic, but whatever. Threats, rituals, sometimes combat, settles disputes. Usually it's mates. Kin selection, when animals wish themselves the same family members. This song just played. Oh, whatever. VLC has a horrible shuffle system. Just to say something random. But, okay. Kin selection, when animals wish themselves the same family members. Altruistic behaviors, increased fitness of genes and close replication. Interesting. Okay. Different species interacting with each other. Community ecology. Okay, so this is where we're getting into symbiosis. This is also something that I just kind of forgot. It's here in our notes. We are still billions away, but hopefully we can speak on this stuff later. So think of it like this. So these basically competition is bad for both of them because they both waste energy in, you know, doing the competition. Competitive principle, exclusion principle, one with slight reproductive advantage will eliminate the other. And then predation. You know, one organism benefits, the predator, but the other one doesn't, the prey, because the prey dies. But then prey do have defenses. So for example, you can see, so wasps are horrible creatures, but then these animals have evolved to look like wasps, which in turn, deter the what do you call it? Deters the predators. And then you have warning colors, Bastian mimicry, so harmless, but it mimics the harmful. And then Malarian. Malarian mimics, looks dangerous, and they are dangerous. I guess because of the stings, but I hate the paper wasp. I hate any wasp. One stung me during golf tryouts, actually. Parrotism. Okay, so you can think of this like a leech. It sucks your blood. And then I don't believe that's good for you. Hold on, let me double check. Leech is bad for you. Okay, so maybe not leeches, but something like that. Like a tick. Tick is a better example. Mutualism. So both species do benefit from the interaction. Symbiosis is not necessarily a bad thing. You could think of venom. The symbiote could either be it lives with you. It's a part of you. What is this? Commensalism. Oh, so you could think of it like I think it's barnacles and whales. So by attaching to the whale, the barnacle, or it's certain types of fish, I think, attaching to a whale or another sea creature, because the other creature doesn't really mind, it doesn't bother them, but then the other one benefits because obviously they're a lot faster, they can pick up the scraps that they don't eat, that the larger creature doesn't eat. Sexual selection. What do you think it means? Promiscuous. You have many partners, and you're showy, and then you don't care for your offspring. Monogamous. You have one part. This is usually what humans engage in. You have one partner. You both have similar structures, and then you care much for your child. I would hope that's how your parents work. Vincerable. Yeah, good job, Malachi. Vincerable. Polygamous. Polynius. Okay, so I guess this is what you need to do. So polygamous is one male and many females, but then polyandry is a showy female. Does that totally make sense? Yeah, I get that. It was the blurst of times. This is Friday Night Funkin'. This one is fire though, so I will keep this going. Keystone species, they're not abundant, but they're very important to the species. They help keep it in check, basically. Invasive species, what do you think it is? It's a species that's invasive. They can enter an environment and then take advantage of its resources, therefore stealing resources from the native species and thus decreasing the total population of the native species. Okay. What did I say? I look you think whatever. Climate, you should know. So climate is just long-term weather. You have macro. So think macro means big, micro means small. Macro is like all the seasons, and the micro is like the smaller areas. Biomes, major type of ecosystems. All right. Biogeography. I think we're about to do an ecological map. So geographical distribution of species. Bio, life, geography. Dispersal, behavior, biotic, abiotic. Alright. Ecology, man. Energy flow. We've already discussed this, but it's the way of energy flows. So gross primary production. So your net primary production is the total amount you make minus the amount you lose. The song's stuck with copyright, so we're gonna move on. Oh, this is the without the lyrics, but it's a Super Mario. Mario teaches typing rap. That's what it is. Here, we'll play the. We'll just wait with the lyric. Actually, yeah, here, take a listen.
Oh, let me show you something right quick. Hold up. See? Yeah.
You got the rap? Yeah, we rapping right now.
Yeah.
Spelled correctly.
Man, Watch this.
Oops. And then net primary production, so it's like I said, so it's the amount you produce minus the amount you lose, basically. Ecological efficiency, what do you think it is? So it's trophic level minus trophic level. If a primary produces this much a day, and this much make it to the primary consumers, what's the efficiency? So I guess think of it like this. It's like, what's the efficiency of the amount of energy that's transferred? Simpson's diversity index. So the diversity equals, it's 1 minus the sum of the total number of organisms minus the total number of all organisms. So organisms of a species and then organisms of all species. So high D value suggests stable and ancient site. So I guess if you think about it, a high D value means that there's a lot of different species, I guess. There's a lot of species, and then not one single species is taking over, because then there's more species, more biodiversity. But then the less that you have, it means that there's maybe like one species there or something. But it says here. And then so this is how you calculate it. All right. These will be on your formula sheet. You just need to know how to use them. Let me think. Exponential growth. The change. Okay. So this is calculus. The change in population size over the change in time equals the rate of population growth times population size, and the growth rate equals births minus deaths over the population, which makes sense if you think about it. You just have to know how to use this. Logistical growth. I'll show you what logistical growth looks like. It's this. So basically, it means there's a carrying capacity. So then, you only have so many resources to go around. And that's what that is. And then, so know that K is the carrying capacity, N is the population size. Hold on, is this equation wrong? Should you have divided by 15? I think you were supposed to divide by 900. Whatever, that's okay. My exponential growth. We hit 8 million. We hit 8 billion people. 2022, I think, or 2023. So we have a lot of people on this planet. It would be really funny if that actually wasn't true. Unlimited resources are rare. Really? Are selected species. Species that exhibit rapid growth, where opportunistic species quickly invade habitats, reproduce, and die. So you could think of it like the if you remember the type of death graphs, it's going to be type 3, where you have a lot of offspring, and then not many end up surviving, I guess. Okay, selected species. Wow, so much Friday Night Funkin' in here. It's where population remains constant and they produce a small number of relatively large offspring. Okay, so then then you can't afford to have that many because they all require a lot of resources each. Prenatal care, over. And then K is carrying capacity. You have your factors, sort of independent, it's independent, blah blah blah. We've already talked about this. You only keep about 10% of the energy of the trophic level above you, and the rest is lost to heat. Let's see. Energy, biogeochemical cycles, life, earth, chemical cycles. Okay, now we're on, so these different cycles, this is what it is. Let's see. We're getting materials available, And then so once these fossilize, you have different layers. So organic materials are unavailable as nutrients. Oil, you can't eat those. But then these are burned. And then so inorganic materials become inorganic materials are available, say water. So through the formation of sedimentary rock, then you have all these minerals. Inorganic materials are unavailable. But then through weathering and erosion, you create all this sort of things. I actually don't know how to explain this. This is not my strong suit. But okay, so the water cycle, right? Hydrologic cycle, whatever. Water evaporates, evaporation, condensation, precipitation, and I forgot what the rest of it was called. I don't know, percolation, evapotranspiration, carbon cycle. So CO2 is removed by photosynthesis because they turn photosynthesis. They turn carbon into they turn this into glucose, or not necessarily glucose, but into sugar, energy, stored energy. The carbon cycle involves recycling Earth's carbon atoms through Earth's biosphere into organisms as carbohydrates and back into the atmosphere as carbon dioxide. At the highest level of organism, carbon cycle can be simplified. Photosynthesis. So photosynthesis, you convert light energy into ATP and NADP. I think it's NADPH. I'm pretty sure, yeah. And then you use those to convert carbon dioxide into sugar. Decomposition into the soil, then combustion is that burning of fossil fuels. Okay, nitrogen cycles. We also do need nitrogen. Nitrogen fixation, assimilation, ammonification, nitrification, and denitrification. I did write it down, but I probably did not know what I was talking about. We're almost done with this. We're about to go to chemistry. Chemistry is my strong suit. Just reservoir. Nitrogen fixation. Nitrogen gas is fixed into ammonia. Ionizes to ammonium by acquiring hydrogen ions. So hydrogen ions. If they're an ion, that means they've lost. They do not have an equal amount of electrons and neutrons, and then so it'll probably have a positive charge. I'm pretty sure. So in this case, by having an additional hydrogen, hydrogen ion specifically, you have a positive charge. Nitrogen fixation, so nitrogen depletes by bacteria, nitrification, ammonium to nitrite, which is observed by plants, and then denitrification is the release of nitrogen back into the atmosphere. Okay. The phosphorus cycle. Weathering rocks releasing phosphate into the soil. Producers taking phosphate, incorporated biological molecules. Phosphate returns to the soil via decomposition of biomass. So we do have we have a bunch of phosphorus. It's called the phosphorus bilayer or phospholipid bilayer, because you have the polar head, and then you have the non-polar tails, which are it's either the saturated or unsaturated, that's bent unsaturated fatty acids, and then those make up our membranes. The bilayer, there's two of them. Let's see. Bioremediation, bioaugmentation. So mediation is the use of organisms to detoxify, and then augmentation is where you introduce species to essentially, I guess the thing is that the difference is detoxify. Human impact, we accelerate changes, makes sense. Let's see. Biomagnification, the process which toxins or pollutants become more concentrated as they move up the food chain. This occurs because animals at the top of the food chain consume many smaller animals and thus have accumulated the toxin, therefore increasing the concentration. Harmful effects. I think I see. Wow, poor humans. Eutrophication, I'm gonna guess how that's pronounced. Process by which a body of water becomes enriched in excess developed nutrients, typically phosphates and nitrates, leading to an overgrowth of aquatic plants and algae. Ramifications, reduction in oxygen levels, algae blooms. Hypoxic water, low oxygen. Hypo, so hypo means less. So you have like, you know, hyponitric acid, and then you have more, so like peroxide. Let me look it up real quick. AP Chem. Per, yeah, it's per. So per means like the extra, hypo means less. Okay, we finished with that. Now we are on to basic chemistry, which I believe was the first thing on here. No, that's the chemistry of organic molecules. We're in chapter 2, basic chemistry. I make chemistry puns periodically. Yeah. Right. Ooh, okay, I'm gonna say a little funny ones, because you can see it says from school. I took a summer school class, so I didn't have to take PE in person. And then this was one of the songs, because we were learning about rhythm. And then, I don't know if I can get Spotify or VLC to do it, but this song is a perfect loop. So like if I play it on Spotify, it'll just loop through perfectly. And it sounds unironically really, really good. Gamsa Bonita. Gamsa Bonita. Gamsa Bonita. Sorry, Instagram reel. Okay, so it doesn't loop perfectly here, but on a software that's better than VLC, even though VLC is really, really good. Whatever. Okay. Matter, anything that has mass. Anything that has mass and takes up space. Element, substance, that cannot be broken down. So think of it like this. You can't break down water. You can't break down hydrogen into 2 more hydrogens. So it's at its lowest point. If you break it, I don't know what separating the proton and neutron does. It's probably not good. I know those are what create isotopes, so I don't know if that actually works for hydrogen. And then so elements make up, they're made up of atoms. So you have, you have the ones that we know about are protons, neutrons, and electrons. Those are all the ones you need to know. There's, there's quarks and then all those sort of things, but yeah. Chnops. So carbon, hydrogen, oxide, nitrogen, oxygen, phosphorus, and sulfur make up 95 of us chnops. The subatomic particles, they're really, really tiny. Also, I lied. These are not atoms. They make up atoms. I don't know what I was thinking. It says elements are made up of a bunch of atoms. Whatever. Okay, protons, pro, neutron, neutral. Electrons are negatively charged. Electrons have electron shells. You probably learned it. So, like, for example, you have this group, then this group, then this group, then SP, then S, or SD, SD, SPD, SPD, S, FP, S, or no, it's, yeah, SPD, S, shoot, wow, I forgot all my chemistry, whatever, that's okay, I'm sure it'll be fine, I can rerun it someday. Alright, moving on, protons, neutrons, both have an atomic mass number of 1, and then so depending on how many there are, that's what contributes to its the number you see on the periodic table, this. But then the number that actually shows up on here is the it's the, what do you call it? It's the average of all of the isotopes that exist, including the proportions, I'm pretty sure. And the atom is mostly empty space. Electrons don't have like significant weight, I guess that's what it is. Atomic numbers, the number of protons in the nucleus. Chemical symbol is one of 2 letters that represent it. So you have like oxygen, Au is gold, Sr is strontium. Mass number, so it's the number of protons and neutrons, so that's the mass number. is the number of protons and neutrons, but the atomic mass, as I said, the average of all the mass numbers for all the isotopes. And then basically what an isotope is if it has the same number, it's like same number of protons, because a proton is what defines the element, but a different number of neutrons. Periodic table. You should know this. So groups have similar chemical properties, and then periods, horizontal rows, the properties change. And then you can tell, like, for example, the more higher up and the more right most, this is more electronegative, which means that it has there's higher energies that pull electrons to it. Except we exclude the noble gases because they have full shells, and therefore they don't really need any more electrons. They can still bond by donating to a different group, but that's different, you know. I think this is an old model. People say atoms aren't important, but they matter. You should laugh. Isotopes, as I've explained, atoms are the same with different neutrons. So, like, see, 6, 7, 8. And then so what you'll do is you take up the weights, so this is 12, 13, 14. You multiply them by the proportions, and then you get the average. And then the 12 is the total number of protons and neutrons. So, like, for example, you'll see carbon. It's really close to 12 because a majority of the carbon atoms that exist have 6 protons and 6 neutrons. Molecules, any combination of elements. Compound, molecules that include more than one element. And then all compounds are molecules, right? But not all molecules are compounds, because you can have, like, for example, O2. That's a molecule because there's 2 oxygen atoms, but it's not a compound because it's only oxygen. And now water, H2O, is a molecule and a compound because it's a compound because there's more than one element. It's hydrogen and oxygen. Molecular formula. I hate the structural formulas, but, you know, you gotta know what they are. So you have molecular formula, which is this. Then you have your molecular structure, which shows how the atoms are bonded together. I don't know what carious means. Oh, this is important to know. The shape of a molecule affects its function. You can see this, for example, in enzymes. Enzymes at their active site, it's a very specific shape to bind to a very specific substrate. And then so if that is changed, whether it's through, like, maybe denaturing or competitive or non-competitive inhibitors, maybe non-competitive bonds to its allosteric site, changes the shape, then it can't function because it can't bond to that specific substrate anymore. So it's very important. Isomers, molecules that have identical molecular formulas but different arrangements of atoms. Okay, let's see what that. Hold on. Iso prefix equal, same, or identical. What? I feel like it's the opposite of here, but whatever. Well, I guess because it's the same. It's the same formula, but then it's different. Yeah. Valence electrons. It's in the furthermost shell. Determine which way the atoms will combine. Okay. Determine number of. Yeah. And then it's the middle is because they're transitional metals, and then so that's not the. So it's a little different. Atoms with 8 valence electrons, even with the noble gases. Atoms can gain or lose electrons to complete this rule. Usually, the first 3, so groups 1, 2, and 3, like to lose electrons, and then 5, 6, 7, like. When did this mute? How long ago did this mute? Hold on. It's a problem when it's been almost 30 minutes. Okay, maybe just a simple tap button was a flaw. So what we're gonna do? There's no way to get around it unless I like flip the inside. Whatever, I'll just have to deal with it, and I'll just have to be careful. That's what the monitoring is for. I noticed it. A little too late, but whatever. Okay, so nucleic acids, blah blah blah. ATP, adenosine triphosphate. Yeah, it's in the name. ATP, so there's triphosphate, and then ADP is diphosphate. And then, so the energy is released once the bond between one of these phosphate groups is broken. And then it requires energy, basically, to put it back together. Alright. Okay, so now we're done with that minute, and we're going to take a break. We're going to make some text specifically for this. Let's see. We're going to call it... I believe 20 is the max. When I say 20 is the max, you're supposed to do it for me. Alright, and then to make this even more immersive, we're going to do put a filter on, and scroll from also, my focus is not cybersecurity, it's AP biology. Okay, we're gonna go take a break. cell structure and function. Okay. So this is chapter 4. This is funny because it's true, right? Cell theory. All organisms are made up of cells. Cells are the structural functional living unit of all things. Cells come from pre-existing cells. So we already talked about this, prokaryote versus eukaryote. So bacteria and archaea are prokaryote, and then everything else is eukaryote. See, no membrane-bound organelles, no nucleus, and they're smaller. They're more simple. But then these, you can see, there's comparative. Once it comes up, I'll be able to read it. And then so now we're just talking about each of the organelles. So these are ribosomes are made up of 2 subunits. It's protein and ribosomal RNA or rRNA. And then they produce proteins based on the DNA sequence, which was based on the mRNA, which actually comes from the DNA. That makes sense. And then you have the plasma membrane. It's the phospholipid bilayer with embedded proteins. So like you have a channel protein, you have a transmembrane protein, and then you have peripheral protein. We'll talk about those later. The cytosol, which is it's the fluid and stuff, water and organic molecules. It holds everything in place within the molecule. And I guess there's a difference between cytosol and cytoplasm. So cytosol is in the cell membrane. Cytoplasm is a cell component present inside. So cytoplasm is a cell component, but cytosol is fluid. Okay. I guess this just helps. They have all of those. Eukaryotic cells. They evolved. Let's see, unfolding. And then so this is where the endosymbiotic theory comes from. Basically, is that at one point prokaryotes didn't have these organelles, but then some kind of bacteria that was basically the mitochondria and the chloroplast in plants. Yeah, they were their own independent bacteria, but then they were absorbed by organelles, and then they it was a huge evolutionary advantage. So those that had them, they would go on to live longer, have better fitness, basically. And then so why they're double membraned is because the outside membrane's from the eukaryotic cell, but the inside membrane's from its actual prokaryotic history. And that's our DNA. There's circular DNA, which resembles the prokaryotic cells. This is evidence for it. Those are called plasmids, I'm pretty sure. And then there's ribosomes, and the ribosomes resemble those. Of these organelles, the ribosomes resemble those of prokaryotes. They're really similar, basically. And then so reproduction, cell reproduction. We're still talking about the organelles within. They divide in an asexual faction, which is similar to binary fission, which is how bacteria separates. They reproduce laterally, or this way. And then similar in size to prokaryotic. So this is just evidence for it, right? See? So mitochondria. And then this is a... what kind of tree is this? Hold on. I forgot what it was called. Let me find it. I don't know, but which is bad. Phylogenetic, that's what it's called. This is a phylogenetic tree. Okay, nucleus, it's found near the center. It contains and protects DNA. It has chromatin inside, which it later wraps around, I forget what they're called, but the chromatin wraps around DNA, and then it creates chromosomes. Let's see, nucleus is a dark region within the nucleus. It creates ribosomes. Sure. Okay, eukaryotic cells. We're still there. So nuclear envelope It's a double membrane found around the nucleus, and the nuclear pores, they're holes. So it's similar to the pores on your face. If you don't know what a pore is, they're like little holes. And they regulate, what goes in and out. Endomembrane system. You have the ER, the endoplasmic reticulum. You have the Golgi body. You have different types of vesicles, and then you have lysosomes and peroxisomes. So we'll talk about these. So, and the endoplasmic reticulum ER, there's two kinds. There's the smooth and the rough. The rough comes from the fact that there are ribosomes on there. So here, see, it's membrane connected to the outer nuclear envelope, and then ribosomes attached on it, which make it look rough. And then so they produce proteins, and then they fold the extracellular proteins inside that. Smooth ER is connected to the rough ER, and the nuclear envelope too, I guess. And they produce lipids, break molecule down. So it's responsible for detoxifying. That's what's important. But then they also send vesicles, which are, think of them like little packages. They send them to the Golgi body. Yeah, Golgi body. See, they're basically like little packages for transport. And there's two ways. We'll talk about like how this actually works later. And then the Golgi body, it's a bunch of flattened vesicles. Sure. They receive the vesicles, they modify, and then they ship them around the cell. I believe they can also ship them outside of the cell. So this is an animation. It basically shows how these exit. Extracellular proteins, they're released outside the cell. Basically, these fuse with the membrane, and then they kind of stretch it out, and then they release it out. Rough endoplasmic reticulum. Oh yeah, you could select it because I did OCR onto this PDF. Oh, excuse me, I'm coughing. Lysosomes, membrane-bound vesicles produced by Golgi body apparatus. No Golgi apparatus. Digestive enzymes, destroy non-functioning organelles, pathogens. It's like a defense, I guess. Peroxisomes, membrane-bound organelles containing enzymes. They help break down fatty acids and dangerous chemicals. Okay. Vacuoles. Okay, so this is specific to plant cells. They store. So you can when we talk about like hypotonic and hypertonic solutions, we'll see more of that come into play. The chloroplasts, again, specific to plant cells. They're filled with pigments, photosystem 1, photosystem 2, photosystem 1. It's the order they were discovered in, but photosystem 2 actually is used first within the light dependent cycle. Photosynthesis, why couldn't I think of the name? And then so within it there are thylakoids, and then stacks of thylakoids are called the granum, and then the stroma is the intermembranous space in between. And then the thylakoid space, that's a little different, that's on the inside. And so we'll talk about that later once we get to photosynthesis. The mitochondria, not the powerhouse of the cell. It's a double membrane because of the based on the endosymbiotic theory. That's where cellular respiration happens. It's in, I believe, both of the organisms, but since we don't have photosynthesis, we can't make energy from light. And then so here, these are just some differences. Plants have the cell wall, which are much more rigid. Rectangular shape, no lysosomes, yeah. Cell wall, this is specific to plants. They're made up of carbohydrates and proteins, and they maintain the shape. It's very strong. That's why it's called a wall, I believe. And the cytoskeleton, this is found in both kinds of plants. Network of protein fibers, they maintain the shell's shape and they assist with movement within the cell. Hold on, let me find, yeah, not microtubular, are they? Maybe it's, let's see. It's that kinesin protein. See, and it looks absolutely hilarious. I'm pretty sure this is how it works, but it's actually hilarious. Like, just look at the way it's walking. See, motor protein. It's. I can't watch comments because it's my school account, but just imagine that it's really funny. Okay. Microfilaments, active 2 chains, structural support. Intermediate filaments. Keratin, static. Okay. Keratin, microtubules, stronghold, consists of proteins of two, function help with the structure of the cell. Yeah. Okay. Haters are going to hate. Okay, now we're talking about surface area, I believe. We're still in this section. Yeah, this is the last part of this chapter. So basically, so in order to be the most efficient, you want a large surface area to volume, as in you want more surface area for the amount of volume. So as you can see here, surface, if it's just a 1 by 1, 1 by 1 by 1, the ratio is 6. If it's 750 by 125, ratio is 6. But if it's, and then this is because, see, this is, instead of just being a 5 by 5 by 5, then the only surface area it's going to be 20. I actually think this is incorrect, because what this should be is it should be 150. No, not 150. 125. Oh, that's the volume, not surface area. I'm sorry, 25 times 6. Apologies. But then you can see it's a lot more inefficient. So you can see basically a large surface area. There's more chemical exchange because there's more surface area to do that chemical exchange. Does that make sense? Okay, ecology, we already did that. And the next chapter is membrane structure. Five. Oh, that's a lot of fives. 5.1. No, 2, 3. Let's see. It appears I missed a chapter. Hold on. I can just talk about what's in my notes. Okay. So we're talking about membrane structure. This chapter isn't that long. It only took about 6 pages. So basically, should really, I'll find the slides. Hold on. Teacher actually uploaded them to, but it's also my fault for not noticing. So here's what I'll do. And then so I'll just explain each of these. Oh, it's this guy. He could also explain it. Well, it's a 5-minute video. But yeah, that's all horrible. Well, these are all not high. Let's see. But okay, so you can kind of see what's going on, right? Okay, so I'll go over these once I find them. Phospholipids, we already talked about those. So species that live in cold environments, they have a higher percentage of unsaturated, so those are the ones bent. And then some species will increase the percent of unsaturated fat during cold months. Because unsaturated it aids in cell fluidity, and I guess that's helpful. So membrane proteins, they have many different kinds of functions. You have cholesterol, which is these, right? And then so basically they help the plasma membrane be less rigid, and it's similar to unsaturated fatty acids, but they're not present in plant cells. Then we have glycolipids and glycoproteins. So right here, and then glycolipids, glycoproteins. And then so what they do, they're carbohydrates that attach to phospholipids and membrane proteins. The immune system must recognize these features for a transplant to be a success. So I guess they're a key part of the cell. As you know, cells make up tissue and tissue make up organs. Then we have the integral proteins, which are these right here. Or no, sorry, these. And then so what they do is they're permanently embedded within the membrane's bilayer. I don't have anything else written for that. Peripheral proteins, so these ones. I believe they're detachable. Yes, they can detach, and they're located on top of the membrane, so they're not with aside from the nonpolar area. You have aquaporins, and those are specific because they allow water to pass through the membrane, because water can't go through the bilayer because inside is a nonpolar, and so they don't, they're not chill with each other. Selective permeability, basically that means that only allowing certain molecules into the cell, keeping others out. Nonpolar molecules cross easily, but polar molecules need energy, or they need to go through a protein. So for example, that's what channel proteins are for. So they allow passing molecules through the membrane, and they go with the concentration gradient. So what that means, let's say out here you have a lot of protein, but here you have a little of that protein. The concentration gradient goes from high concentration to low concentration, so it's going to naturally want to diffuse downwards into the area with less concentration. We'll talk about that more once that comes up. See, carrier proteins, cell recognition proteins. Let's see. Okay, so cell recognition proteins help immune system, diaries. I have no idea what I wrote. My handwriting is illegible. Receptor proteins, area where, it's like similar to the nerve cells we were talking about. But then they also aid in cellular communication. We'll talk more about that later. Enzymatic proteins and enzymes within the cell membrane. I don't see any here. Junction proteins, proteins that communicate with other cells. I'll pull up a picture of that one, because I do remember that being mentioned. I remember this song. Let's see, it's like this. These junctions. And then they aid with cellular communication, I guess. Then you have the extracellular matrix as a meshwork of proteins and polysaccharides located outside the membrane. I'll show you that here. Alright. So like you can see here. They aid with communication, and they connect to the cytoskeleton through the membrane proteins, and they hold tissue together. So I guess that's how cells become tissue. Integrin, transmembrane proteins, links ECM and cytoskeleton. Collagen, it gives the matrix strength, so it's itself a protein. Apparently, around 25 to 30% of all protein is collagen. Interesting. Desmosomes, they're connections that hold cell together. Spot welds. Oh, this is the photo that my teacher showed me. See, we have all that. Tight junctions, membrane proteins that attach to each other. They create watertight seals, like bladder and the kidney. Oh, so I guess they're able to open up when needed. Gap junction, located where water and ions can travel through. Plasma what the freak? Do the slides. Do we have hypotonic? Dang. Okay, so we're really just missing all those. Alright. Alright, now here's so I just explained what the concentration gradient was, but now we also have diffusion. So it's the movement of molecules from high to low concentrations. That's a concentration gradient. But then facilitated diffusion, no, that's different. Facilitated diffusion is across through channel proteins. So it needs help. Those are usually the larger molecules. Osmosis is the diffusion of water across a selectively permeable membrane from high concentration to low concentration. Molecules don't cross the membrane. Okay, hypotonic solution, isotonic, and hypertonic. Let's do those. So, hypo versus hyper versus iso solutions. Okay, so isotonic, it's where the cell is, basically the amount going in is the same as the amount going out. Hypertonic is where more water is leaving. So you can think of it as if the concentration of water outside of that cell was lower than the concentration of water inside the cell, so the water diffuses out. And then hypotonic is it's basically the opposite. So let's say there's more water outside of the cell than on the inside, and so it'll diffuse in, and so the cell will bloat. We still have more. So plasmolysis, when the cytoplasm of a cell shrinks due to osmosis. Plasmolysis. Oh, okay. So this is specifically for plants, plant cells. So you have shrinking of the. So turgid and plasmolysis. Flaccid. So flaccid is isotonic, plasmo is hyper, and then turgid is hypo. Okay. I could roll with that, I guess. Okay, let's see here. Exocytosis, endocytosis, phagocytosis. Okay, I should show this. Well, they're pretty self-explanatory. I'll just explain them. So you have protein pumps. And the protein pumps use ATP to move molecules against the concentration gradient. So, for example, if you want to go from a low concentration to a high concentration, and then so it requires energy to do that. That's ATP. Exocytosis, basically, is where an intracellular vesicle it fuses with the plasma membrane and it secretes the substance out. That's what we saw before. But then endo is where it breaks off a part of that and then becomes a vesicle, so it enters the cell. And then you can think phagocytosis is where a cell takes in a large substance, and then pinocytosis, you think pino is like high, pino kind of sounds like small, micro. It takes in a small substances. Alright, water potential. This I do need to show. Alright, so you can think of water potential with this formula right here, and then there's the solute potential, which is a part of it. So water potential equals the solute potential plus the pressure potential. You'll be given the equation for these in the equation sheet. And then, so this is what it is. So the solute potential is basically negative the number of ions. It's usually going to be 1. And you have the molar concentration, the pressure constant, and then the temperature in Kelvin. And then so what you can think of it as the more solute that a solution has, the lower its water potential is going to be. And therefore, the way water potential works is that water moves from a high water potential to a low water potential. Let's see. Just looking through here to see if I need to say anything else. So you can think of it, a higher water potential to a low water potential is the same thing as a low solute concentration, because with a low solute concentration means that there's going to be a higher water concentration, and you're going to go from a low solute to a high solute because it's going to be low water to, or high water concentration to low water concentration, and then it's also high pressure to low pressure. It makes sense if you think about it. And now we can go to cell communication. Cell communication and signaling. What about this for about 2 hours? Actually, I think what I might do is I might continue this later. So I am getting a little sleepy. And I think I do have some time tomorrow for the bio test. So, and I don't have any more finals. Let's think about it. I have AP Biology. I finished AP Psych, and I finished AP Biology. I have some AP Cybersecurity stuff, but it's not going to be that hard. AP Cybersecurity is good. Then AP, let's see, just looking at all my classes. Spanish final, I just need to make my presentation look good, basically. No, actually, I just need to make a presentation with photos and then write on the note card. It shouldn't take too long. Then we have AP Stats. We did the final already, we're just getting ready for the AP test. Then, let's see. AP Psych, I just finished my final. I'm done with AP Psych. See, AP Biology, literally good. We're about to take the test. Not doing anything Monday or Tuesday. Tuesday or Thursday, I should say. And then band is nothing. And then I'm up to date on my CE classes. So actually, I think we're doing pretty good. So I don't have much to worry about. Yeah. So we're gonna be stopping. We're gonna be taking our break. So I think we got a lot of work done. I would say 2 hours. I'd probably get rest for 15 minutes or more.